So first of all, there are 4 processes that are required for life. And those processes, first of all, are going to be growth. Organisms have to be able to grow, and our cells and tissues have to be able to grow too. We have to be able to reproduce if we're going to be considered living. So an organism would have to be able to produce new organisms like itself, or a cell would have to be able to produce new cells like itself. Responsiveness is a third one. This means responding to the environment, responding to other cells, so responding to stimuli in some way. And then lastly, in order to be considered alive or living, we have to be able to complete metabolic reactions. These metabolic reactions will fall into different categories, and we'll talk about those more, but we do have to be able to build molecules and then also take molecules apart. So with that in mind, the cell is considered to be the simplest structure of matter that can live. So it's able to do all of these 4 functions that we've listed here. It can grow, it can reproduce, respond, and then it can also run metabolic reactions. So this is a look at the cell here. You can see there's a lot of different parts and pieces to the cell, and we're now going to begin our investigation of those different parts. So if we talk first about general features of cells, these would be things that you find in most cells. Specifically, these are going to be in eukaryotic cells, which will be more complex cells, which are the type of cells that make up the human body. The first one to talk about is the nucleus. And the nucleus is considered to be the most conspicuous organelle. And what we mean by the most conspicuous organelle is this is the most obvious organelle. It's the easiest one to see. And in the diagram up here, it is the purple structure that you see in the middle. And then if we look at this electron microscope picture, we can see this dark spot in the middle of the cell, and that would be considered the nucleus. Now the nucleus, its job is the control center of the cell. A lot of times we think of it as being comparable to the brain because it is essentially running the different reactions that are occurring inside of the cell. It's controlling all of the activities that would be taking place. The reason why the nucleus is going to be able to control all the activities inside the cell is because it does contain the majority of the genetic information. So in other words, it contains the DNA or the chromosomes. And this nucleus is going to be enclosed by a nuclear envelope. The nuclear envelope is a membrane barrier. It's going to be around the outside. And one of the unique things about the nucleus's membrane is going to be that it's actually a double membrane. There's 2 layers of membrane material that are going to compose that nuclear envelope. So the function of the nucleus is, first of all, to store all that genetic information, and then second, it's going to use that information to control the cell, and the way that it will control the cell is that it does control protein synthesis, or in other words, the making of the proteins that will be involved in all of the different reactions that the cell performs. If we do take a closer look at the nucleus, because it is such an important feature in a cell, the nucleus, we can see it closer in this picture here. It is the purple structure. And one thing we've already mentioned is that it does have this nuclear envelope, which is that big membrane around the outside, double membrane around the outside, which will really isolate the internal components, which is the DNA, from what you find outside of the nucleus. Now, it's such important information inside of the nucleus. It is the blueprint to do everything that the cell might need to do. So we do have to have a way to access that information. And that's what the nuclear pores are going to do. The nuclear pores are just little holes or tunnels that run from outside the nucleus to the inside. And this will allow certain molecules to enter and exit the nucleus. And this, again, will be responsible for that control of the cell's activities. The nucleolus is the dark spot that we often see inside of the nucleus. This is going to be where the ribosomes actually get put together, or where they do get assembled at. A lot of times we see more than one of them. It's a dark spot. There might be 1, 2, 3, something along those lines. In this particular picture, we just see one nucleolus. And then the final thing, which again we've already mentioned, is that there is going to be DNA there, but that DNA happens to be wrapped up with a lot of proteins. So when we talk about the DNA plus the proteins and we put it together, we use the term chromatin to describe that. So the nucleus does contain chromatin. So the nucleus is running the cell. That's kind of the brain, if you will. There are, though, a lot of other features for the cells that we do want to introduce here. One of them is the plasma membrane. The plasma membrane is this barrier that goes all the way around the outside of the cell. So it is the exterior of cells. It is a selective barrier that allows sufficient passage of oxygen, nutrients, waste. Basically, it regulates everything that gets to come in and everything that's going to go out of the cell. So it is controlling entry and exit is the way that we say that. If we talk about what the plasma membrane is made of, there's two main types of molecules that are holding this together. We have phospholipids, and then there's also going to be proteins. And if we talk about the phospholipids first, these are the main structural part of the membrane. So in other words, they really hold it together. Those are these little molecules down here. Notice that they are drawn kind of like this. And you'll have a whole bunch of those that are going to join together side by side. And then we'll actually have two layers of them. And this is what we call a phospholipid bilayer, and bi because there are two layers of them oriented in kind of opposite directions. That's the main structural part of the membrane. And then the proteins are going to provide what I like to describe as the functional part of the membrane. That's what will make one membrane unique compared to another membrane. And the plasma membrane will have proteins embedded all throughout it, and those are the purple structures that we see in this picture. And then in addition to that, you can very well have some sugars and things on the outside. So we see those. Those are important a lot of times for recognition. So they help cells to recognize each other. They're also going to be important for communication purposes. We talk about that entry and exit function. The main purpose of the plasma membrane is to regulate what enters and what exits. And it's called selective permeability because it is actually selecting what gets to cross and what does not. And what does cross and what doesn't is going to vary from day to day or moment to moment. So we do have certain molecules that are not able to regularly just get across this membrane. And those types of things are going to be primarily large substances. So if they're really big, they would have a tough time squeezing through. We're going to have ions and other charged substances that have difficulty getting through. And that will be just because of the chemical nature of the inside of the membrane prohibits them from crossing. And then other hydrophilic substances are not going to be able to cross the membrane. Now, sometimes the cell definitely needs some large things to cross, or some ions or hydrophilic substances to get across. So the way that they will actually help those get across or select them for crossing is going to be the proteins. And that's why I describe the proteins a lot of times as a functional part of the membrane, because they get to make those determinations. So the embedded proteins, they will assist with the substances crossing, and the things that they would be assisting with would be all of these that can't get across on their own. If we talk about how the cells are going to actually be held together, because humans are made up of lots of cells, and those cells are making up our tissues and our organs. Well, one way that the cells are going to be held together is they can be held together by a type of glue. And that glue is going to use glycoproteins, sugary proteins on the outside. Those proteins are part of what we call the glycocalyx, and it's just going to kind of hold the different cells together. We can also have cells that actually fit together as kind of a tongue and groove fit or a puzzle-like fit. And then another way is that we have special junctions between cells that help to hold them together. These fall into different categories, and this image right here is showing us some of the main junctions that we do find between human cells. And the first ones are the tight junctions. The tight junctions are kind of a waterproofing seal around cells. They are going to completely encircle the cell, and then they bind them together in a watertight fashion. Another type is the desmosomes. Desmosomes are for really anchoring or tying cells together so that they are hard to pull apart. And then the third one that we want to mention here is the gap junction. Gap junctions are for communication because they will allow molecules to actually pass from one cell to the next. And so these tiny little communication tunnels are really going to help the cells to respond to each other and whatever happens to be their current situation. So those are our cellular connections. Just inside of the plasma membrane, we have what's called the cytoplasm. The cytoplasm is a cellular material that basically is anything in between here and here. So we're not talking about the nucleus, but we're talking about everything outside the nucleus until we get to the plasma membrane. And that's a lot of different materials. The materials that we do have in the cytoplasm fall into three main categories. We have the cytosol, the organelles, and then there's also inclusions. The cytosol is a fluid that is the water-based material that we do find outside of the nucleus. There's water there, and then there will also be a lot of things dissolved in the water. So other nutrients and molecules, maybe some sugars, that would be there. The organelles we're going to get into later because those are the larger structures that you see here in all the different colors, and they perform a lot of different activities and roles for the cell. And then the inclusions are going to be chemical substances that you just find kind of in droplets or granules throughout the cytoplasm. So these might be lipid drops, they might be glycogen granules, so that's a polysaccharide that we would find there. And again, those come in a variety of different forms, and they would be present in the cytoplasm. If we take some time now to talk more about the organelles, like I said, they're all these different structures that we're seeing here. There are many of them, and each one of them, as you can see already, they look a little different. They have different sizes and shapes, and they will have different roles that they play for the overall cell. Organelles are considered little organs, so that's where the name comes from. They are specialized compartments. Most of them have their own membrane around the outside. Since it is a membrane, it is going to be made up of the phospholipids and the proteins, just like the plasma membrane was composed of. And they're not only isolating that compartment, but they do also regulate what enters and exits that particular little organ. The first one to talk about is the mitochondria. The mitochondria, as you can see here, is kind of a long, slender organelle. It looks a little bit like a little worm, and the mitochondria contain two membranes around them. So they have a double membrane similar to the nucleus. And these membranes, the outer one is kind of just a normal membrane that goes around the outside like this, but the inner membrane, if you look, it just folds in all over. So we have these little finger-like projections, and those finger-like projections are called crista or cristae. These are going to greatly increase the surface area of that particular membrane, which is really important for providing energy for the cell. The mitochondria's main job is to be the power supply or the powerhouse of the cell because it will provide most of the ATP that the cell uses for its various metabolic reactions. Another thing about the mitochondria is that it does contain a little bit of DNA, and it also contains some of its own ribosomes. The ribosomes, to talk about them a little bit more because we did just introduce them, the ribosomes are very tiny structures. They actually don't have their own membrane around them, and these are going to be contained in the cytoplasm. Some of them are just floating around in the cytoplasm. Some of them are actually going to be stuck onto some other organelles, and we'll see that in a moment. They are larger than what we're going to find in the prokaryotic cells, so larger ribosomes than you might find in, say, a bacterial cell, and their main job is to make proteins. We will have lots and lots of these inside of a single cell because cells do need lots of proteins in order to run their various activities. One of the largest organelles that we do have inside the cell is what we call the endoplasmic reticulum. And while that's quite a mouthful, we just abbreviate it the ER, and we call it the ER. And the ER is a very large network of membranes. So it is very massive inside the cell. It is a bunch of membranes that are really folded back to create all of these small, slender compartments. And we do divide the ER into two main categories. We have what's called smooth ER and then rough ER. And they look a little different and they have different overall functions inside the cell. The rough ER is rough because there will be little ribosomes stuck on the surface. And so that is what we see here that looks like it's spotted. The spots are the ribosomes that are stuck on the surface. And the ribosomes are responsible for making proteins. So this part of the ER does have some role in protein production, and it's also responsible for making the majority of the cell's membranes. The smooth ER, on the other hand, doesn't have the ribosomes on the outside, and that's why it's smooth, and its job is more lipid metabolism. So making of a variety of different lipids such as steroids and fatty acids, and it also is going to play a detoxification role. So it will help the cell to get rid of various poisons and toxic molecules. If we look closer at the ER here, the rough ER specifically, we can see what that ribosome on the outside of the ER is actually doing. So the ribosome is the brown structure. The ribosome is making protein, which is shown here in purple. And as the ribosome makes the protein, it actually takes that protein and sticks it into the inside of the rough ER. And then the rough ER is going to take it and it will package it up into this little compartment. And so this compartment right here is what we call a vesicle. It's a little bubble, really, of membrane on the outside with our protein packaged in the middle, and that is for transportation. So now that that particular protein has been packaged up in this little compartment, that vesicle can be sent somewhere else inside the cell where we can do something else with it, or that protein is going to be able to complete its specific function. Another one of the organelles that we have is called the Golgi Apparatus. And the Golgi Apparatus is usually pretty close to the ER. And it does a lot of times receive and take in those little packages that the ER is sending out. So the Golgi, we think of it as the processing plant of the cell because it's going to receive what the ER sends, it's going to process those materials, and then it's going to repackage them and send them out. So if we kind of tie it all together and look at what the Golgi is doing, the blue picture is showing us the rough ER. And the rough ER is taking the proteins from the ribosomes on the outside, packaging them up in those little vesicle compartments, and then sending them out. And we can see a number of these kind of being sent out. Those are then going to travel to the Golgi, shown here in green. The Golgi receives them and takes them in. It processes the contents of those, modifies them a little bit if need be, and then it's going to pinch off again so that we've got these proteins which are ready to go, packaged up, and they're going to be then sent out into the cell to whatever their final destination happens to be. In some cases, we're actually sending them to the outside or the exterior of the cell, and they would then basically dump the contents to the outside, so they would be secreting them, is the way that we would say that. In other cases, they're actually going to stay inside and they may travel to some other compartment like we see up here. We have talked now about a number of these different compartments or organelles that we have in the cytoplasm of the cell. Just a few other ones to mention that we haven't discussed specifically. The lysosomes are responsible for digestion. So that is their main job, to digest different food molecules that we might need to take apart to get some energy out of. The peroxisomes, if we find the peroxisomes on here, those are similar in their overall appearance, but the peroxisomes do have a special role in detoxification. And then we also have some generic compartments called vacuoles. And the vacuoles primarily are for storage. That could be storage of food, it could be storage of waste, really storage of whatever the cell happens to need to store. Each one of these compartments is going to have its own role that it plays. Ultimately, they're all working together to run that one cell, but they've just somewhat subdivided the labor that has to be completed.